Hello, my name is Nicky Craig and I would like to introduce our letter to the editor entitled Reduced IL-31 receptor alpha splice variant messenger RNA following allergen challenge in a canine model of atopic dermatitis. The cytokine-specific interleukin-31 receptor alpha comprises a signaling peptide, 5 fibronectin type 3 domains, of which the first 2 include the cytokine binding domain in the human, a transmembrane domain, and an intracellular region. In humans, at least 12 IL-31 receptor alpha isoforms are described, of which some are functional, some have reduced function, some are non-functional or inhibitory, and some are soluble. In contrast, dogs have only 3, of which 2 are full-length and assumed to be functional isoforms, and 1 is a short isoform which is missing the signaling peptide and the first fibronectin type 3 domain. This isoform is assumed to be non-functional. In this study, we used RT-qPCR to investigate transcription of IL-31 receptor alpha long and short isoforms in the skin of atopic laboratory beagles over the course of acute allergic inflammation, following chronic allergic inflammation, or in the skin of normal dogs. We found that during both acute and chronic allergic inflammation, transcription of both long and short IL-31 receptor alpha isoforms was reduced in the skin of allergic dogs. We suggest that this may be a mechanism to limit IL-31 signaling in the presence of inflammation in the skin. In contrast, in the absence of allergic inflammation or following resolution of an acute flare, the non-functional short isoform was consistently transcribed at a higher level than the long isoform. This was also the case in normal dog skin. We suggest that this may be a separate mechanism to modulate IL-31 signaling through post-transcriptional regulation of the receptor in normal skin. In conclusion, regulation of IL-31 receptor alpha transcription and alternative splicing to a presumed non-functional isoform are potentially 2 separate mechanisms by which IL-31 signaling in the skin may be differentially regulated during allergic inflammation or in its absence, respectively. Thank you for your attention.